Wonderful. Okay, this meeting is being recorded. I consent to that. Okay. Hello, everyone. Welcome to PSPlatypus. I'm Kyle, and today I'm going to be taking you through homeostasis and acid-base, and also central dogma. Lovely. Okay, well, let's get on with it. All right. It's only a few slides, but the slides are a bit packed. Okay, so the normal pH in the body is 7.4. So you'd think, oh, that's not neutral, but actually, generally what we need is a specific pH for protein folding. And so the normal pH of the body is 7.4, although in different compartments you'll find different. For instance, in the stomach, it's much lower, more acidic. So homeostasis is the steady keeping of conditions inside the body. So really, we don't want things to be massively changing. We want to keep things steady. And that doesn't mean keeping them the exact same everywhere. It just means keeping a general control of the body, basically. So yeah, as I was saying, pH is extremely important because the number of hydrogen ions, the concentration of hydrogen ions, that will affect how proteins fold. And the folding of proteins, as you may know, secondary, tertiary, and quaternary structure all contribute to the actual functionality of a protein. So you can see here, this is just an example graph, and you can see the optimum pH of a specific enzyme is here 8. And as the pH deviates from that, it gets less and less active. And so it's very important that the body keeps the pH around 7.4, plus or minus 0.04, to make sure that proteins can still function. So in practice, although Professor Chris Wright would argue that there is a slightly more nuanced explanation to it, but in practice, medical practitioners will say that someone is in acidosis when their pH is below 7.4 or 7.36, and they're in alkalosis when their pH is greater than the normal pH, so 7.44 or above 7.44. And also acidemia, Chris Wright, he uses this interchangeably with acidosis. So I think for all intents and purposes, it's okay to consider them the same generally. I think technically acidemia is the state and acidosis is the process, but people just say acidosis. And the concentration of water in the body is 55.5 molarity, and it's a constant. Okay, the next slide. Here we are, we're talking about partial pressures now. So partial pressure, so that's the pressure of the gases in question. So for instance, with the air that we're breathing, hopefully, the total atmospheric pressure is 760 millimeters of mercury, and air is about 21% oxygen. So the partial pressure for oxygen is 160 millimeters of mercury. And so CO2 is what we expire, right? We breathe in oxygen to do all of the good energy stuff, and we breathe out carbon dioxide as a waste product. And so carbon dioxide, if dissolved in the body's water, can actually produce carbonic acid, which you can imagine would increase the number of hydrogen ions and thus the acidity of the body, right? Lowering the pH. So if you're not breathing, right, then you're not breathing out carbon dioxide. And so that causes an increase in carbon dioxide inside your body compared to normal, where it's being breathed out. That increases the amount of hydrogen ions that are inside your body, which lowers the pH, and that's called respiratory acidosis. So the normal partial pressure of carbon dioxide in the body is 36 to 44 millimeters of mercury. And you'll realize that actually, there's a nice memory aid where that actually connects with the normal pH being 7.36 to 7.44, so 36, 44. So it's a nice memory aid. They're not actually connected though, at least not directly. But yeah, so that's the normal partial pressure of carbon dioxide in the body. And so if a patient had an ABG taken, so that's where you measure the gases in the blood, and their pH was 7.05, remember, much lower than the 7.36, and the partial pressure of carbon dioxide was 80 millimeters of mercury, so that's higher than the 44, that would be indicating respiratory acidosis. So that's the body, because of respiratory problems, like not being able to breathe, is increasing the acidity inside the body. Yeah, and that's obviously not good for the functionality of proteins and thus the well-being of the patient. And this is the final slide on homeostasis and acid-base. So I'll note on neutrality. So while we say that normal pH is 7.4, and that's where we have the acidosis and alkalosis coming from, there is actually slightly more nuance that Chris Wright goes into in his lecture. So at laboratory temperature, pH 7 is neutral, right? However, at body temperature, 37 degrees Celsius, sorry, 6.8 is actually the neutral pH. And that's because as temperature increases, water actually dissociates into hydrogen and hydroxide ions. And so because of that, you're actually getting more hydrogen ions, right? Because water is dissociating into the acid and base products there. So you can see, because pH is actually hydrogen ions, the amount of hydrogen ions is increased because the temperature is increased, causing that dissociation. So the pH is still neutral, however, because it's dissociating into equal amounts of hydrogen ions and hydroxide. However, there's more hydrogen ions in the solution. So the pH is neutral, of a 6.8 pH is neutral at 37 degrees Celsius. So that's a bit more. So I went through it quite quickly, but I tend to do that. So please let me know if I'm going too fast or too slow. Sorry.
When it refers to just pH, just of the patient, like where specifically do you measure the pH? Because you did mention that different departments we have different pH levels.
Yeah, I think we're talking about the extracellular fluid. Right. But I'll clarify that. I'll revise the slides when I'm done with that. And I'll make sure. So when you go back to the slides, perhaps in a couple of days, that'll be clearer. Sorry. Thank you. Anything else? Okay, well, I'll move on then. Oh, is everything still going? Yep, okay, good. Sorry, just make sure that my computer hasn't crashed. Okay, so central dogma. So this is the big one. Okay, so central dogma overview. So the central dogma is the general idea of how genetic information is expressed into proteins. So how genetic information is actually carried out in biological systems. And very generally, it follows the flow of DNA to RNA to protein. You can see also, so we've got DNA to RNA, that process is generally called transcription, and RNA to protein or polypeptides is translation. And we've also got here this nice arcing arrow back to DNA, and that's DNA replication. So every time a cell divides, it needs to create another copy, right? So that both cells have a full genome. Okay, so, and feel free to interrupt at any point because I can't see any faces, so I can't gauge the excitement or the comprehension of the group. So just unmute and yell out, and I'll stop, I'll slow down. Talk about any specific topic with you. Okay, so DNA structure. So it's a double helix, which means two spirals with anti-parallel strands, which means that the two strands, they run in opposite directions, and it has a uniform diameter, which is what it sounds like. The width is consistent along the length. So it is composed of a deoxyribose sugar phosphate backbone. So the sugar is deoxyribose, hence the D in DNA, and nitrogenous base, which is actually what is holding the encoding information. Now you've got covalent bonds in the nice backbone, as you can see here. You've got a zoom in of the sugar phosphate backbone. You've got the backbone here, and then you've got the bases in here, and they're all complementary. Oh, well, they're not all they're complementary to their pair, so adenine to thymine and cytosine to guanine. But you can see we've got covalent bonding here along the backbone, but then we've only got hydrogen bonding between the base pairs. So that enables DNA to unzip and connect back together, sort of, whereas the backbone provides this strong structure so that the sequence is maintained. And the way that we determine the directionality of the strand is by counting the carbons in the deoxyribose sugar. So we count them from the nitrogenous base. So 1', 2', 3', 4' and 5'. So generally we look at 3' and 5' because that's where the rest of the backbone is connecting. So you can see you've got the 5' end of this strand, and 3' would be more down here in the other direction, and 3' on this end. So you can see how they're anti-parallel. They run in opposite directions. Okay, so this structure facilitates the essential functions of DNA. 4 essential functions. So those are to store genetic information. It's pretty important that DNA stores genetic information. That it replicates precisely, and that's so that you carry the genetic information to the progeny of the cells. Because if you don't have precise replication, then you're not really doing the job that DNA should be doing. It needs to be readable for gene expression because, well, why are you storing this genetic information? It's so that it can be expressed. But also, in relation to that precise replication, it also needs to be somewhat susceptible to mutation. If it was completely unsusceptible to mutation, then there would be no opportunity for mutations. And as you might know, mutation is how you may randomly get some genetic diversity that may actually give an advantageous trait through evolution. And you can see the structure here. The key points here are that there are 4 nucleotide bases, which facilitate for the code. The bases have hydrogen bonds, which enables sort of the unzipping for replication for gene expression. You've got a phosphoribose backbone, which enables the storage of the genetic information in a relatively strong way. And you've got base complementarity and the complementary nature. So if there's an adenine on one strand, then the complementary base will be a thymine on the other strand. It means that you can take one strand and read it, and you'll be able to predict what the other strand will say. And that's how DNA replication happens. I see that there's some messages in the chat, so I'll just have a quick look. I'll quickly jump back to homeostasis then. Actually, I'll return to homeostasis at the end of this, just so that I'm not jumping around, because I've just gotten a question about the homeostasis part. But I'll get back to that. Okay, so DNA is organized in the cell. Basically, it's very long, right, to record all of the information that makes up an entire organism. You're going to need a lot of genetic code. And so you have to coil it many, many times upon many, many times so that you can fit it in a small volume. So it's stored in the nucleus. It's tightly coiled around histone proteins. So you can see sort of this nice image from Wikipedia that has sort of progressive zooming out as the DNA is coiled more and more. So around the core histones, histone H1, but you don't need to know this in great depth. But just giving you an idea that it's very tightly coiled so that you can actually fit the DNA inside of the nucleus. So it's coiled over and over and zooming out and coiled more, and you've got scaffolding proteins in there as well. Okay, so now we're at the beginning of DNA replication. So it's making copies of DNA. And the start, it makes sense to start at the start, the origin. So the origin of replication is where replication originates. It's where DNA polymerase binds. And in prokaryotes, you can see some prokaryotic DNA here. It only has one replication site, or one origin of replication. And so because of that, you can see it'll take a bit longer, but it will eventually get all the way through. But in eukaryotes, which are humans, coincidentally, we have multiple origin sites. So eukaryotes have multiple origin sites, and that enables one, as I was saying, the really long strands of DNA to be replicated more quickly, because I think it would take something like 8 days to replicate, whereas it's much quicker with with multiple replication sites. But also very important, right? If the prokaryote loses this one replication site, this replication doesn't have anywhere to start, right? Whereas if a eukaryote loses an origin of replication, then it's okay because eventually you can imagine one of the other ones will catch up to it, right? So we've got this nice evolutionary adaptation that avoids the complete loss of a chromosome if we lost one origin of replication. So we have many. Okay. So these are the steps of DNA replication, and I'll just go through them quickly because you can always read the slides in your own time, and I don't want to bore you or confuse you too much. But basically, you have an enzyme called DNA helicase, so that binds to the origin and that unwinds DNA, and that breaks the weaker hydrogen bonds between the base pairs. And you've also got single-stranded binding proteins, that's SSB, that keep the strands separated temporarily, so they don't immediately just zip back up. Okay, then you've got primase, and that attaches RNA primers near the origin, and that's showing, hey, DNA polymerase. And so that's how DNA polymerase knows where to start, and that's where it starts from. DNA polymerase is the big deal. So DNA polymerase binds to the primer, and it's got a sliding clamp that's keeping it in place. I'll show you a diagram just after this. And then it runs in a 3' to 5' direction, joining complementary bases and writing in the anti-parallel direction, 5' to 3', so the opposite direction. And that's because it's making another copy. So it's going to make copy. And so I have a nice little mnemonic for this. I'm not sure how much other people will like it. I quite like it. So it's a phonetic mnemonic. It's based on sound. So DNA polymerase reads 3' to 5', right? It runs along the template strand in a 3' to 5' direction. And then it writes the new DNA, the coding strand, the new coding strand, in 5' to 3'. Writes 5, reads 3. I don't know. I quite like it. And that's just for polymerases. Okay. So you might be thinking, oh, well, if there's RNA primers attaching to the start, right, then that RNA primer will actually be taking up some of the place of where the polymerase would be writing on. So basically, another enzyme called RNase H removes the primers later, and then DNA polymerase can fill that space up with DNA. And there's another enzyme that comes and fixes the backbones because there's actually a little gap in the sugar phosphate backbone that needs to be linked up. So that's done by an enzyme called ligase. On top of that, you can imagine DNA can only run. Well, actually, I'll get to the leading and lagging strands in a second with diagrams. It'll be a bit easier to understand. Okay, here we go. So here's our beautiful DNA replication. I've got more chats. I see, okay. Is the leading strand always the parent strand of DNA? That's a good question, Ben. I'm thinking about it. And really so on any strand, it's just that the leading strand will be the strand that you, actually, I'm not too sure. I'll have to remember that question and get back to it. Sorry, Ben. It's hard to think right now, aside from what I'm exactly saying, but sorry, I'll get back to you, Ben. Sorry for the eclectic presentation. Okay, so here you have the diagram. You've got DNA polymerase. You've got the sliding clamp behind it. So where were we? So we've got the helicase that unzips. Then we've got the primase that is putting the RNA primer so that the DNA polymerase knows where to start. And then you've got DNA polymerase running off from the primer and creating the new strands, right? Now, what we've got here is because DNA polymerase reads 3' to 5', right? It'll be able to, as helicase is unzipping in the DNA, it'll be able to run smoothly in one direction, but in the other direction, because it runs 3' to 5', right? 3' to 5'. On this side, it will be running across, but then helicase will unzip some more, and then it'll have to put another primer, and then it'll have to do it again. So it does it in a much more fragmented method. And those are called Okazaki fragments. And those Okazaki fragments are what compose the lagging strands. The lagging strand, because it's lagging behind, whereas the leading strand can just be done in one nice long process. Okay, here we go. Okay, so I'm just going to talk about the where you actually get the energy, because for any reaction, right, you need energy to go in to be able to be creating bonds, right? So the energy for DNA synthesis is provided by the cleaving of pyrophosphate, right? So the breaking of a bond gives the energy to create a new bond. So what we've got here is actually the building blocks, right? We've got the nucleotides, which are the bases and their sugars and phosphate. And what we've got here is they actually start off as a deoxyribonucleoside triphosphate. So there's actually 3 phosphates attached originally. And those extra 2 phosphates provide the energy when, see, for instance, cytosine is pairing with guanine. We're able to predict that, oh, because it's guanine, well, cytosine is the complementary one. So that's going to bind to it. And so as DNA polymerase is coming along, it takes the energy from the phosphate ion and it breaks and it attaches cytosine to continue along the writing of the growing strand. And the pyrophosphate is broken off, and that's giving the energy for the reaction. Yeah. Okay, so leading and lagging strands. So as I was saying, so DNA polymerase, it reads 3' to 5', back to that rhyming mnemonic I'm so fond of. So the leading strand is going to be continuous as the parent DNA is unwound. So if you're looking at it, you can see it's got just one nice continuous strand running 3' to 5' on the template strand. Well, sorry, 3' to 5' on the strand. And then you've got the leading strand is just going smoothly 5' to 3' being written. However, the lagging strand, it has Okazaki fragments as a result of it running in the opposite direction. So as it's unzipping, right? See, it just has a nice place to start and it can continue. But you can imagine as we unzip more, we're going to need to have another primer to start from. So it comes in these fragments. And then after that, as I was saying earlier, these primers have to be removed so that we can actually put DNA back there because we don't want RNA, we want just DNA in the DNA replication. So RNase H will remove the RNA primer and then polymerase will replace it. And then the backbone gaps will be linked by ligase so that we've got a nice strong sugar phosphate backbone with those covalent bonds, and that's continuous because otherwise it would sort of start falling apart. For a similar reason, because we have polymerase running reading 3' to 5' and writing 5' to 3', actually when we run along, right, eventually we're going to have overhangs, and that's because of the same idea as with the Okazaki fragments. Basically, we're not going to have anywhere to put the final primer, right? So that we can write along, and then where those primers are, we won't be able to create DNA. So the way that we actually fix the fact that otherwise the lagging strands can't be replicated is basically to solve it, we have an enzyme called telomerase, and telomerase extends the lagging strand, and that prevents degradation. Because if you think about it, if this end went shorter, then we just have this end just floating out, right, of the lagging strand. And so the end of the lagging strand would be hanging out there, and that would slowly be degraded. And you can imagine it's sort of like if you think of a rope and you say didn't have a knot at the end of it or didn't have a nice cleanly cut end, it would slowly fray away. And so actually, after many DNA replications through cell division, we would actually lose all of the genetic code to a point where the cell would become senescent, and it would actually just stop dividing because it would realize, oh, we don't have the correct information and we're running low on the genetic code that we need to replicate. So to fix that, we have telomerase, and that basically fills in these overhangs. Okay. So with that precise replication, there are 3 types of repair systems. You don't need to know much detail about them, about as much detail as I've got written here. So you have proofreading, which occurs during replication, and basically, I'm pretty sure is done by DNA polymerase, but don't quote me because it's not necessary to know. But yeah, that basically is checking, okay, have we actually got a cytosine? Okay, well, that means that we should have a guanine. Is a guanine actually there? Yes. Okay, good. We're business as usual. Oh, is there something that's actually wrong? Then we might need mismatch repair. So the guanine is not there. It's actually cytosine. And then for some reason, adenine, the A, is there. And that's not actually complementary. So it goes, oh, well, there's an error here. Okay, well, we'll take that out. We'll fix it. And that occurs with the main thing to know about these 3 systems is when they occur relative to replication or cell life. Okay, so now we're on from DNA to RNA. Okay, so back to DNA. So you have deoxyribose, right, which is deoxy, so no oxygen on, or no hydroxide on the, or alcohol group on the 2' carbon, right, in DNA. You have uracil instead of thymine. So instead of a T, there's a U, which is a slightly different base, but it still pairs with adenine. Talking about RNA, you have 3 main types. So you have messenger RNA, transfer RNA, and ribosomal RNA. Yeah, RNA is more transient, so it doesn't last as long as DNA because it performs different functions to DNA, and it also isn't proofread. So because RNA polymerase doesn't proofread, whereas DNA polymerase does. Okay, role of RNA in DNA in gene expression. So RNA is integral to gene expression. It's very, very important. So you have different types of RNA. So messenger RNA, that's this one over here, it performs different key functions. It delivers genetic code. So it is transcribed from DNA. It's basically the messenger. It's transcribed from DNA and it's sent from the nucleus to ribosomes, which basically are the machines that build proteins from the mRNA code. Ribosomal RNA is a component that makes up the ribosome, which is that factory that makes proteins. And transfer RNA, it delivers the building blocks of proteins to ribosomes. So you can imagine, very important in converting DNA into protein, RNA. Okay, so the transcription process, it's a bit long as well, but it has a similar concept to DNA synthesis. It's just that it's not for replication, it's to be transcribed to then be converted into protein, to have some sort of effect inside the cell. So RNA polymerase, it binds the promoter sequence. It doesn't need a primer. And there are these things called transcription factors, which I'll talk about soon, that also affect binding. And the promoter sequence is what tells the RNA polymerase which strand is the template strand. So for different genes, different strands will be the template strand. But the promoter sequence is how RNA polymerase knows. RNA polymerase unwinds the DNA, so it's actually quite impressive. It doesn't need helicase or anything. It unwinds it itself. It reads the template strand. Once again, reads in 3' to 5', so it's consistent for both polymerases, DNA and RNA. And it writes in 5' to 3'. Although, note, instead of thymine, right, it's got uracil. And it continues until it reaches a termination sequence, at which point it stops eventually. And we've got 3 different types of polymerase, and in eukaryotes, generally they produce the different types of RNA. But that isn't very high yield. And in prokaryotes, you've got one type of RNA that produces all RNA types. And what were you saying? Oh, yes, returning to the mnemonic reminder. So yeah, reads 3' to 5', writes 5' to 3'. Okay. So transcription factors, as I was talking about in the previous slide. So transcription factors, well, factors in biology are generally proteins, and these ones are related to transcription. So the process of making mRNA from DNA. So transcription factors, they affect transcription by binding to nearby DNA. So we've got some diagrams here, this one of an activator and this one of a repressor. So activators bind to the enhancer site, which is somewhere else on the DNA. And they also bind to the promoter site. And generally they make a bridge, which actually helps RNA polymerase find where to start transcribing. It's a bit hard to understand, but you don't need to know it in detail. But yeah, it makes a bridge between the enhancer and promoter sites, and this curve, for whatever reason, it helps RNA polymerase find where to start transcribing to start making mRNA. Repressors sort of do an opposite thing. So they muck around with these activators and they prevent transcription. Yeah. Okay, so here we have just a diagram. And this is where it's quite relevant to think about the coding versus template strand. Those are the terms that we use. So the template strand is the one that RNA polymerase will bind to. That's the one that the promoter sequence will be on, where it says, oh, come here, start transcribing. Now, it will be complementary, right? So it sees a thymine there, so it makes an adenine. It sees an adenine here, and it doesn't make a thymine, it makes a uracil. So that's very important to remember. It's uracil, not thymine in RNA. And what it will do, you can see it's writing 5' to 3', reading 3' to 5'. But the key thing here is names, because they might trick you, well, not trick you, but you might get confused in questions. So the template strand is what is read, and it will be and the mRNA will be complementary to that. So that means that we actually get the coding strand and the mRNA, or the pre-mRNA, they will be the same, aside from the uracil versus thymine and then being RNA versus DNA, but they'll have the same sequence. And so that's quite important to remember. If it asks you, oh, this is the sequence on the coding strand of DNA, what will the resulting mRNA strand read? You would actually go, oh, well, it's the same, except that instead of thymines, it's uracils. You wouldn't go, oh, well, actually, I remember that they're complementary, so I'll switch them around. That's not how it works. RNA is the same sequence as the coding strand, but with uracils instead of thymines. Okay, I'll speed up a little bit because I feel like I'm running a bit slowly. Okay, so RNA synthesis processing. So once we've made our mRNA, right, we've made the sequence. Then we need to process it to actually turn it from what is technically pre-mRNA to mRNA. It's pre-mRNA because it doesn't have the processing yet. So post-transcriptional modification. So to turn pre-mRNA to mRNA, it involves adding a methyl G cap or a methyl 5' G cap at the 5' end of the RNA, and a poly-A tail of the 3' end. And that's to basically prevent degradation or slow down degradation of the RNA. So it isn't just immediately broken down because it has, the loose ends, those loose strings, sort of like on the DNA. And the other thing that it has to do is it has to splice out introns. So we have something called SNRPs, and they basically join and together they form things called spliceosomes. And those are what splice out the introns, right? So that we only have the exons. The way to remember which one is the one that is expressed, well, exons are expressed and introns are actually, that comes from intervening segment. It's the ones that sit in between, but actually aren't in the final mature mRNA. So we have pre-mRNA or something just in the middle of pre-mRNA here, but we've got the poly-A tail, we've got the 5' cap. And then through splicing, then we get to the mature mRNA after processing. Okay. Oh, there's lots of questions. I see. Oh, okay. Thank you. Thanks, James. I think you're answering the questions. So if there are any more, I'll address them and I'll send something out so that people have answers. Okay, here we go. All right, so now we're up to protein synthesis. We've dealt with transcription. We've dealt with the modification that goes after transcription to actually make it into mRNA. So there are 4 key steps in translation. So that's loading up the tRNAs, the tRNAs being the molecules that bring the amino acids, the building blocks of proteins, to the ribosome so that it actually has something to build the protein out of. Then we have initiation, which is the beginning of translating the mRNA into the protein. Elongation, which is the process of actually making the length of protein. And termination, which is, okay, we've read all of the mRNA, we've made the protein, now it's time to stop. And that will end up with the polypeptide. So step 1, charging of amino acids onto tRNAs. So basically, there are different tRNAs for each amino acid. And there's something called aminoacyl TRA synthetase. Basically, that attaches the amino acid, the building block, to the tRNA being the thing that carries the building block, the amino acid, to the ribosome. It's attached to the 3' end, as you can see in this diagram. And basically, the tRNA synthetase, they are unique to each tRNA as well. So there's a tRNA, for instance, for serine and a tRNA for leucine, and for different each amino acid, there's a different tRNA. And the tRNA on the other end to the amino acid has something called an anticodon. So the anticodon, if it'll go away here, sorry, the slide thing is in the way. Well, I'll just wait for it to go out of the way and I'll leave it, sorry. Okay, so you can see on the green sort of here with the circle, that says anticodon. So that's the part of the tRNA that actually recognizes the mRNA and goes, okay, well, this is the sequence GCC. So we're going to have CGG, and oh, that actually corresponds to a specific amino acid. Yeah, someone unmute. Okay. Then we have initiation. So this is where the good stuff happens. So you have an initiation complex that's the tRNA and its amino acid and mRNA on the ribosome. Anyway, very complex, but basically the mRNA meets the ribosome and it binds to the 5' end of RNA where the actual reading begins, and it actually goes along 5' to 3'. So it starts at the 5' cap, remember, on the mRNA. And the start coding, AUG, for methionine, designates the first amino acid. So that methionine isn't always inside the end protein, but basically it says start here, and then it continues through. And you have a process directed by these things called initiation factors. And you use guanosine triphosphate to actually pay the energy for the process. So I'm about to get to a diagram, and actually I'll jump through, but this is just a larger version of it. It's just zoomed out here. So here you can see we've got the ribosome, the large ribosomal subunit, small ribosomal subunit, that's the aqua here. The purple is the mRNA, and the red is tRNA, and the pink is the amino acids. So basically, it's a cyclic process, right? Because it's bringing in each tRNA, and that's adding one more amino acid, and it's doing that over and over and over to make a nice long chain of proteins, of amino acids to make a protein. So we have, basically it's you can see here the aqua ribosome. It is running from left to right, 5' to 3'. And so you can see that the mRNA would be going through it right to left. So the first thing it encounters is the A site, or the amino acyl site. And that's where the tRNA will start to bind. And that's where it actually starts to read. And then it gets, so you have binding of the tRNA. And the tRNA read the mRNA by directly connecting, as I was saying, with the anticodon and the codon and going, oh, it's complementary. So yes, this is the correct amino acid coded for by this triplet of the mRNA. So you have the contact point. The anticodon is complementary to the mRNA codon, and that they interact in an anti-parallel orientation, as you'd expect with nucleic acids, RNA and DNA, or RNA and RNA, sorry, here. Okay, and then you have the formation of the peptide bond, and that's in the P site, the peptidyl site. And that's actually where you can see the chain of amino acids is elongating, and then it moves along more. The ribosome moves along a bit more, and that's when you get to the exit site. So A for aminoacyl, that's where it starts to bind and read. P for peptidyl, where it's actually creating the peptide bond between the amino acids, and E for exit, so where it's leaving the ribosome, because the tRNA will have dropped off the amino acid and will have to go away to get recharged, as I was saying in the first step back a while ago, the charging of amino acids onto tRNAs. And then, yeah, you have the release of the tRNA without its amino acid. And that cycle repeats and repeats and repeats for every single amino acid. Okay, and then finally, when you get to the end, you'll have a release factor terminating the translation. And that's basically when you get to a stop codon, so that's UAA, UAG, or UGA, that reaches the A site, so the first site that the mRNA and tRNA encounter. There's no tRNA for the stop codon. So that instead of a tRNA binding to it, what you get is something called a release factor, which is another protein. And that causes the ribosome to disassemble and disassociate from the mRNA, because you've got the protein in its entirety, polypeptide. Okay, now we're on to gene expression regulation. So we've just gone through translation. Now we're going to talk about a few ways that the cell controls how DNA is expressed. Yeah, and this is more low yield, as James has just reminded me. So I'll just go through it very quickly. So one thing you have is called DNA acetylation. So as you may remember, we had the DNA wrapped up in histones, and that's to help it coil it and keep it all nice and small so that it fits inside the DNA. But also, it controls how accessible it is, right? If you're able to walk up to, I don't know, a poster and read it, that's going to be great. But if you have a book that's locked in a briefcase, that's locked in a safe, that's on an island that is surrounded by an ocean infested with sharks, it's going to be harder to access it, just because there's so many things that are sort of blocking you from getting to it, right? So it's a similar idea here from my off-the-cuff analogy. So you have heterochromatin, meaning different chromatin. Basically, so you have the histone tails, they interact with the negatively charged DNA because they're positive, so they're attracted to it. Heterochromatin is basically when the histone tails are tightly wrapping it, as you can see in this picture here. And that means the DNA is sort of locked up. It can't be transcribed because polymerase and all the other enzymes can't get to it. Polymerase and all. So because each chromosome has a distinctive shape, when it's called heterochromatin because you might have seen a karyotype before, and that's where you have all of the chromosomes lined up, including the X and Y and the 23 somatic chromosomes. They all look different. And so it's all really tightly packed up when you actually karyotype so that you can see the differences. So it's called heterochromatin. Euchromatin, good chromatin, U meaning good. It means it's accessible, it's easily transcribed, and that's when acetylation, so when an acetyl group binds to these positively charged tails, reducing the charge because acetyl groups are uncharged, and they loosen up DNA. And so histone acetyltransferase is an enzyme that attaches to these tails. So you can see it's zoomed in here, got it wrapped up, and basically when you acetylate the histones, then the DNA is more loosened up. You haven't got the tails tightly wrapping around it. It's more loosened up so that it's accessible and can be transcribed. Methylation, a similar idea. It only happened, well, actually not super similar, but similar name. So it's the addition of a methyl group to the pyrimidine ring of cytosine molecules specifically. So the cytosine, not adenine, thymine, or guanine. It doesn't actually affect base pairing, but it prevents the genetic transcription. So it's another way of turning off gene transcription, but it does it in an epigenetic way. So it's not actually changing the code, it's just sort of locking it out. Different cell types have different patterns of doing this sort of methylation. And when they're disrupted, this can contribute to cancer and expression of some diseases. RNA silencing, even more low yield, but basically you might, as DNA is complementary to DNA, or the anticodon to the codon, tRNA and mRNA, you've also got other sorts of RNA that are produced, and they can be used to basically bind to that mRNA, right? And then the mRNA won't go into a ribosome, right? So that's a way of basically saying, oh, well, we've actually produced mRNA, but we want to stop it. And that can be used in different therapies and other processes. But yeah, very simply, just have a look at the slide. It's pretty low yield. Yeah, siRNA is more specific. I remember that because S specific and microRNA is less specific. So siRNA is specific to like one sequence. It will bind only to one sequence, whereas mRNA can target a bunch of different genes that have been transcribed and stop that mRNA from getting into the ribosome, and tell the ribosome the mRNA to actually be broken down rather than be transcribed into a protein. Another way that we can control things is through something called protein mislocalization and sequestration. So those are actually two separate concepts. So sequestering means like sort of putting away in a separate spot, or separating. So sequestration is actually the way that the body can basically, once you've produced a protein, put it in a compartment where it doesn't do anything, sort of lock it away in a certain cellular box. And then when you actually need it, you can bring it back out and it'll be in a place where it can actually do stuff. So you can sort of control it, not use it until you want it to be active, and then activate it by removing it from that compartment. Mislocalization is, I think, more of an error, and it's when basically the protein is put in the wrong spot. And so correct folding and stability of the protein, being that it depends on pH and other environmental surroundings, as you may remember from the homeostasis part, it changes the folding, so it changes the functionality, and so it can actually get toxic if it's in the wrong compartment. And one more, so we've got post-translational protein control through modification. So basically, you have to, once you've actually made the sequence from translation, remember out of the ribosome, we've got termination, we hit the stop codon, all of that. Then we've got the sequence, right? But we need it to have different other processes to fully make it into the right folding and everything to make it an actually active or functional protein. So there are three examples here. So you've got phosphorylation that is adding phosphate groups, glycosylation, glyco meaning sugar, silation usually meaning addition of a group. So phosphorylation, same idea. But glycosylation is addition of sugars, and that can be involved in things like blood types are determined by the sugar groups on the proteins. And proteolysis is the chopping up of a protein that can affect folding and activation. So you don't want insulin active in the body the whole time. So you've got this pro-insulin, which is sort of it's before active insulin. And then you have some enzymes here that come and they chop off this gray part, and it results in just the sort of the green and the blue, which is active insulin. And that is central dogma for you. So you've got DNA to RNA to protein, and then slightly more detailed layout here. But yeah, we've gotten from one very simple explanation of it to a much more complex idea. But I hope that I've been of some use. So what is a currently active chromosome? So yeah, that's sorry. Sometimes when I say things, I make it sound like it's a term that is generally used, but James has answered here. So James has answered a bunch of the questions. Does anyone have any other questions about central dogma? Feel free to unmute and ask me, and I might be able to I might be able to answer you. Okay, well, on that note then, oh, I'm seeing one is appearing in the chat. Sorry, let's lose some time. Yep, okay. I think that's just James answering more. Yeah, so currently active chromosome is just one that is being used right now. It's one where I just mean like the gene that we're transcribing. Okay. Well, then it's up to cellular energetics and fuels, if I'm not mistaken. Is that correct, James? Sorry, and please forgive me. This is my first time presenting for the PSB, so I'll learn a bit and be a bit better for you guys next time, hopefully. Thanks so much, though.
No worries, man. You did great. And yeah, we have cellular energetics and fuels next up. Whenever you're ready, Mama.
Yep. Just share the screen. Can you see the screen? Sure. All right. So let's begin talking about cellular energetics and fuels. So firstly, we can define like energy have multiple definitions, but energy in biochemical reactions, we can define it as the capacity to change. And it can be either potential energy or kinetic energy. Potential energy is the energy of the state or position. However, kinetic energy is the energy of movement. And we can convert kinetic energy to potential energy and potential energy to kinetic energy. And then we have the laws of thermodynamics. These laws are universal, so they apply to all matter and all energy. The first law of thermodynamics states that energy cannot be created or destroyed. What does that mean? It means that when energy is converted from one form to another, the total energy in the first form and in the second form are all the same. So there is no change. The second law of thermodynamics says that when energy is converted from one form to another, some of that energy becomes unavailable to do work. And this is because of disorder. In other words, we can say that there is no 100% efficient energy conversion. Why is that? This is because of the entropy, which is a measure of the disorder in a system. And if we talk about the metabolism, so there are two aspects of them: catabolism and anabolism. Catabolism is breaking complexes or large molecules into smaller molecules. This process releases energy and it's considered as exogonic reaction because it releases energy and it's spontaneous. Anabolism is the opposite, where it uses precursors or small molecules or simple molecules to synthesize macromolecules. It requires energy, so it's endogonic reaction and it's non-spontaneous. You can remember these two terms by like catabolism from letter C, cut, and anabolism, A, add. And so we're generally talking about the energy and how cell can obtain energy. So we must talk about ATP, which stands for adenosine triphosphate. It's an organic compound that provides energy in the cell to drive other reactions happen in the cell. So the hydrolysis of ATP releases energy, and the forming ATP requires energy. And there's a term called coupled reactions. So when ATP is hydrolyzed, it releases energy. This energy is used for another reactions that need this energy. So we call them coupled reactions. Enzymes are proteins that used to speed up reactions by increasing the rate of reaction, they lower the activation energy. This is the activation energy without using enzyme, and this is the activation energy when we use enzyme. Sorry. So we can see the difference in there. It offers also an easier path for the substrates, and it brings the substrate or reactants closer together. The rate of the chemical reaction is independent to the free energy. However, it's determined by the size of the activation energy. There are like generally two factors affecting the activity of enzymes: concentration of substrates, like the more substrates we have, the more enzyme activity. However, there is a maximum velocity for that, and when we reach that point, it means that all active sites are occupied in the enzymes. And also we have regulatory molecules. You don't need to memorize all of them, just to understand that these regulatory molecules, where some enzymes require them to function. So we have prosthetic groups, inorganic cofactors, and coenzymes. These two permanently will be permanently bounded to the enzymes. However, coenzymes are not permanently bound. And prosthetic groups, coenzymes are non-proteins, inorganic cofactors are proteins. Sorry, also all of them are non-proteins. And if we talk about the regulation of gene expression, it might result in more or fewer enzymes being synthesized, or it can be result in some enzymes that will lose the ability to catalyze or cannot be regulated at all. And there are also enzyme inhibitors, so we can classify them into two groups: irreversible. Irreversible, that means when they bind to the active sites, they will bind permanently, and the enzyme will no longer be a catalyst. However, in reversible inhibitors, we have three groups: competitive, where the inhibitor binds to the active site and preventing the substrate from binding; uncompetitive, where it binds away from the active site. However, it prevents the releasing of the products. And non-competitive inhibitors, where they also bind away from the active site and change the structure of the enzyme, preventing the substrates from binding. And now we will move to fuels. So the main fuel sources in the body are carbohydrates, lipids, and proteins. All these three have different pathway to reach acetyl-CoA, and then the acetyl-CoA enters the citric acid, and then oxidative phosphorylation, which are common among these three. They just differ in how they reach acetyl-CoA. So carbohydrates will reach that by glycolysis, lipids by beta oxidation, and proteins by amino acid catabolism. So we will talk first about the glucose metabolism. There are two main sources of glucose. The first source is through our diet, and the second source are the glycogen that is available in the liver and muscles, which need to be converted into glucose 6-phosphate. In the liver, it will be used to maintain the blood sugar. However, in muscles, it can be directly used for muscle contraction. There are two pathways, depends on the presence of oxygen, aerobic pathway and anaerobic pathway. We'll talk firstly about the aerobic pathway. So the first stage is glycolysis. It takes place in the cytoplasm, so it can happen with or without the presence of oxygen. It has 10 reactions. You don't need to memorize all of this, just understand the key features. Generally, during glycolysis, it will oxidize the 1,6-carbon molecule into 2 3-carbon molecules, and we call them the pyruvates. We can divide these 10 reactions into 2 phases: preparatory phase. I don't know if I'm saying this correctly, but it involves the first 5 reactions where 2 ATP molecules are being used in these reactions, specifically in reaction number 1 and reaction number 3. And then the payout phase, which consists of the last 5 reactions, where 4 ATP molecules are produced, 2 in the reaction number 7 and 2 in the reaction number 10. So the overall output, 2 pyruvates, 2 ATP, and 2 NADH. Then we'll, if there was no oxygen or not enough oxygen, we'll move to lactic acid cycle or Corey cycle, where pyruvate will be converted into lactate and NADH will be converted into NAD plus. And the second reaction is critical for the glycolysis to be continued. However, if we have sufficient oxygen, then we will move to the next reaction, link reaction. Well, basically, we move from the cytosol to the mitochondria. In this reaction, pyruvate will be oxidized and converted to be acetyl-CoA. So, as we said, this occurs in the mitochondrial matrix. And the overall output of each pyruvate, 1 acetyl-CoA, 1 carbon dioxide molecule, and 1 NADH. Then acetyl-CoA will move to citric acid cycle. There is also another pathways, but this is the only pathway if we need energy. So this is an 8-step process to produce carrier molecules, NADH and FADH2. So the main purpose of citric acid cycle is to produce electron carrier molecules, because we will need them in the next stage, which is oxidative phosphorylation. It occurs in the matrix of the mitochondria. Our overall outputs for each acetyl-CoA will have 3 NADH, 3 FADH2, and 1 GTP, which is 1 GTP, which is effectively an ATP and 2 carbon dioxide molecules. Also, one important point we need to understand is that we need high concentrations of NAD plus during this phase because we will use them to produce NADH. Then we'll move to the last stage of producing ATP, which is oxidative phosphorylation. It has two parts, the electron transport chain and chemiosmosis, or we might call it ATP synthesis. And the aim is to produce ATP molecules. So during the electron transport chain, which is the first part of the oxidative phosphorylation, we have 4 complexes here: complex 1, complex 2, complex 3, and complex 4, besides cytochrome C, which is a small molecule, small protein, and ubiquinone, which is type of lipids, I think. So what basically happens, the first complex will reduce NADH to NAD plus, transferring the electrons to complex number 3, and the second complex will reduce FADH2 to FAD, and transferring the electrons also to complex number 3. Then cytochrome C transfers electrons from the third complex to the fourth. Finally, the fourth complex pumps one more ion, hydrogen ion, and the electrons is finally accepted by the final acceptor oxygen, forming water. So complex 1 and complex 3 and 4 are the ones that pumping the hydrogen ions against the concentration gradient. So they pump the hydrogen ions from the inside of the inner membrane into the intramembrane space. And then we'll move to the last stage, which is the chemiosmosis, right, or ATP synthesis, where we have ATP synthase enzyme. This enzyme we can consider it as a channel for hydrogen ions to be to flow down their concentration from the inner membrane space. So it transports the electrons down their concentration. The movement will make this enzyme move. So we can say it will be like a motor, and this movement will phosphorylate ADP and produce ATP. And at the end, we will have, this will produce about 28 ATP molecules. And at the end, like in total, this process will produce 32 ATP molecules. That's it. I hope I was clear. If you have any questions, feel free to ask.
All right, seems like we don't have any major questions that I can see. Feel free to chuck those in the chat at any point and we'll get to them at some point. All right, so our next topic is the medical interview. This is part of clinical skills. Hopefully you've had a little bit of that already. Actually, just chuck me a like a thumbs up reaction if you've done the medical interview, at least the early stages in clinical skills so far. Okay, good stuff. So you guys kind of know this a little bit. Let me share my screen. 1 moment. Okie dokie. Can you all see that? All right, perfect. Okie dokie. So this is essentially clin skills. for your semester. The medical interview is by far the majority of what you'll be doing in clin skills. So this is our attempt to try to condense essentially all of the stuff you'll be doing into one little bit. So it's a big topic, but I guarantee you that it'll be easier and easier and more natural as you go. So just keep that in mind. You don't need to get it straight away. You'll get lots of practice throughout the semester on how to do this. So this is like the medical interview. The purpose of it is basically triaging a patient, trying to figure out what's wrong with them, how serious is it, and then what can we do for them, among other things, a little bit of background information, which is useful to them. So we've got our 6 little steps on the left there. So we've got an introduction, which is in which we sort of do our little admin stuff. We talk a little bit to the patient. Then the big thing is the history of presenting complaint, which you'll probably hear a bit about. The presenting complaint is basically what they came here. So like what prompted them to come to the doctor. And so we want to get as much information about that as we can, because that's probably going to be the most pressing issue. And then the 4 after that are sort of background history. So what are things that we want to know about the patient because it might be relevant to their condition or might help us make a diagnosis. So these are some sort of general tips. So these apply at any point in the medical interview, more or less. So you want to ask open questions because you want to encourage the patient to volunteer any information which might be necessary. So if you ask the patient like closed questions, then you're possibly going to be ending up like where you ask them idea of such and such. Do you have something and such? Do you have a stabbing pain here? If they say no to all of these and they have something else which they know is an issue, but they weren't able to tell you because you didn't ask the right question, then you fail to get that information. So you want to ask open questions in general, and then you ask follow-up questions, like basically based on what popped up during the initial questions that you want to follow up on. And yeah, listen, so let the patient talk. They generally won't throw at you like patients who ramble on and on and on in your OSCEs or like your clinical skills exam, like assessments. So like just listen, let them tell you the stuff because it'll usually all be useful to you. Of course, be respectful and supportive. Summarize back. So this is usually done at the end of the medical interview, but you can also do it at intermediate points if you feel it's necessary. So you basically, you look at the patient, you say, okay, so you've told me blah, 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 blah. Is that all correct? Is there anything I've missed? And that's a really good way to make sure that there's an understanding between you and the patient about what's going on, which is quite important. And this last one is from experience. So it can often be very natural for you, like you're doing a task, you want to find out a certain bit of information, and once you do, you're like, yay, I've done it. And you'll say just on your autopilot, like awesome or perfect. So don't do that. Things like thank you or something like that is generally a good neutral thing to go with, because patients will tell you stuff which is, not good for them. And so you need to both acknowledge that information, but you need to do that in a respectful way, in a way that doesn't make them think, okay, so why is it good that I have diabetes? That's the sort of vibe we're going for. All right, so this is not super relevant for you if you're doing it over Zoom, but once you do it in person, it'll be important. So basically just think about it. And then once you do it in person, you'll be doing this for real. So you want to read through patient records because that's always important. There could be some really big important stuff there. You want to collect any necessary equipment, especially when you move on to examinations in semester 2. Turn your phone off. That's self-explanatory. And hand hygiene. You will hear about hand hygiene a huge amount in clinical skills, because it is quite important. I'm sure COVID has shown us all how important it is to wash your hands. Okie dokie. So this is a big block of text. I'm sure it seems very scary right now, but I guarantee you by the end of semester, you'll be able to do this by memory just immediately. It's just a big block of text. Basically, nothing about it changes from patient to patient. So like you can basically reel off the same thing every time. And so we have a few things we want to do. We want to show who we are because legally we need the patient to know who we are. So I would say, my name is James Gunasegaram and I'm a first-year medical student from Monash University, if I was one of you guys. And then we need to tell them what we're going to do. So I can do that right at the start, and I can do that again later on. So I need to say I'm taking a medical history. And I also want to explain a little about that. Technically, if you think the patient might be familiar with a medical history already, you can probably say, are you familiar with this? And then if they say yes, you don't need to explain it. But in OSCEs, they'll always ask you to explain it. So you might as well just start explaining it. we need to talk about confidentiality. That's a legal requirement. The patient needs to know where this information will be going. So we're held to the standards of any other medical professional. You can tell them that. And the big thing is anything that they tell you will be confidential between you, the patient themselves, and the treating team. So that's their GP, their specialist team, whatever it happens to be. And consent. So consent doesn't mean you need to ask explicitly, you know, do I have your consent or something? Although you can do that. Generally, you want to say, like, you know, is that all okay? Once you're sure that the patient understands what's going on. We'll talk a bit in med law about why it's important they know what's going on. So one thing you might notice here is that we have a little break here, which is where we ask for the patient's full name and ask them what we would like, what they'd like to be called. So there are two reasons that we interrupt this little spiel for this. The first reason is that you may sometimes get the wrong patient. And it is a bit awkward if we go through the whole spiel, we talk about all this stuff, and then we ask, oh yeah, by the way, who are you? Oh, sorry, I'm meant to be taking the patient in the next bed. So we want to establish right at the start, have we got the right patient? And that's why we ask for the full name. And we ask, what would you like to be called? Because some people don't want to be called by their full name. And so we want to be respectful there. Okie dokie, ID data. This is admin stuff. You need to hit on every one of these in your OSCEs, obviously, to get your full points. So age and date of birth, save yourself some maths, like don't ask the date of birth first, because then it's a bit awkward if you ask the age. They'll just say, okay, well, you can work that out, right? So save yourself some maths, ask how old they are first. You can write that down and then say, okay, thanks. What's your like explicit date of birth? And that'll narrow it down a little bit for you. So ask those in that order. And then occupation. So we don't want to assume that they have a current occupation. This is like an ongoing thing. In general, you don't want to assume something about the patient, because then, you know, you're making them feel bad for really no reason. So you say, like, have you got a current occupation? They might say they're a student. They might say they have a job. They might say they're retired. They might say they're unemployed. All of that's fine. And ATSI, so Aboriginal or Torres Strait Islander status. So they'll give you a little phrase which you need to say verbatim for legal reasons. So for us, it was, are you of Aboriginal or Torres Strait Islander origin? It'll probably be the same for you guys. It might be a little bit different. Right, so history presenting complaint, this is the big one. So we want to start with an open-ended question because, well, the patient has come in, they probably have a decent idea of why they came in. So we want to ask him that question. So like, what's brought you in today? And so we ask that first open-ended question. We follow it up with more open-ended questions. We want to let them get as much, we want to get as much as we can just straight up, because it is so much easier to get that information by just tell me stuff rather than asking them specific questions, which will take you more time. So we want to ask them those open-ended questions. Once we're satisfied, we've got all the information we can get from that, then we move on. So we have a little mnemonic here, which is WWQQAA plus B ICE. So the first six are basically our traditional, like, you know, what's important about, sorry, what are the like main characteristics of the disease? And then B ICE is more about the patient themselves. So where is usually for things like pain or sensation. So like, where are you feeling the pins and needles? Where are you feeling the pain? When, so when did it set on? And the next slide we'll talk a bit more about the specifics of when, because that can get really important. The little characteristics of how it set on, is it getting better or worse, all of that sort of stuff. Quality is like, what does it feel like? So you want to avoid asking them specific questions, because if you ask them like, oh, is it a stabbing pain? And they're, you know, they're going to defer to you a little bit when you're, even when you're a medical student, and even more when you're a doctor, because they're going to think like, oh, well, he said it's a stabbing, it probably a stabbing pain. Yeah, I think it sounded like a stabbing pain. And you don't want to do that and then find out later on, well, it felt like something different, but they just thought it was close enough. So you want to ask an open question, let them come up with the initiative and describe something which it feels most like to them. The quantity, if you've ever been asked by a doctor, how does it feel on a scale of 1 to 10? This is that. Make sure you tell them which one is high and which one is low. So like, you don't want to give them an ambiguous thing. The patient gives you a 9 and you're like, oh, well, that's fine, that's nothing. When really they thought the 9 was the top. And in general, we usually put 10 as the most pain. So like 1 is barely noticeable, 10 is the most pain you imagine, something like that. Alleviating aggravating factors. So make sure you ask these separately. So like, ask first, is there anything that makes it better? And then ask, is there anything that makes it worse? You'll get more of the latter, like you'll get more things that make it worse, go figure. But do make sure you ask them separately so that the patient knows what's going on. And associated symptoms. So like, is there anything else out of the ordinary? So we want to think about things which are linked to the presenting complaint. So things which have changed recently. That's the important stuff. All right, so the B ICE part, this is sort of a more modern edition, and it's basically like looking at it from a more patient perspective, we want to know what the patient actually wants. So B's belief, so what do they think it might be? If they come in complaining of like chest pain or something, and you find out they had the same thing 3 weeks ago and it was diagnosed by a doctor, then we've saved ourselves a lot of time. So it's important to ask the patient, what do they think it is? Impacts, this is like how it's impacting their life so far, which is good because it gives us more avenues on how to help them. Concerns, so what are you worried about, basically? This links in a lot with impact, but it's basically like, what is the patient concerned about for the future of their, for the progression of whatever this condition is. Expectations, this is another big one. It's like, what do you want us to do? Because if the patient wants something and then you sort of paper over that for whatever reason, and then that can be quite bad for their impression of you and of the hospital or clinic. So in terms of like actual application to your real job, WWQQAA, essential. Beliefs, quite important. ICE is, it gets a bit muddy because at some point, 90% of what you'll hear is like impact, it hurts or something like that. Concerns, it's going to hurt more, or it might be a heart attack, or it might be cancer. Expectations, I want to get better. So at some point when you like go out into the real world, it can start blending together a little bit. But for uni stuff, Monash stuff, you want to make sure you hit all of those points. WWQA plus B ICE. So yeah, so these are a few more like little details about some of the questions. So the WW, we want to know if there's pain, does it go anywhere? So if you've got pain in your chest, it can mean different things if it's going towards your stomach, or different things if it's going towards your side or towards your neck. So we want to make sure we ask that question. And then when, we want to know if there were circumstances at the start. So like if they have pain in their right arm and it happened right after they fell on their right arm, that's very different to having random pain in your right arm. Sudden or gradual, so did it happen immediately, or was it like a slow fade in? And that's again quite important. All of these things are important to our diagnosis. Is it getting worse, is it getting better, or is it stable? Which can be, especially if it's getting worse, then that can be quite important for us to know. Is it constant or intermittent? So is it like we have a constant pain, or do you have pain sometimes and it goes away and it comes back, blah, 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 blah. And if it's intermittent, if it comes and goes, then how often does it come and go? Oh, I already mentioned alleviating and aggravating factors. Okie dokie. So a medical history of a few things here. So we want to know any current medical problems, because these can be quite relevant. Any past medical problems as well, because if they had something in the past, that might have flared up again. Any surgeries and any hospitalizations. So if they've ever had a medical procedure, or if they've ever had to be an inpatient stay, so like they're actually admitted to hospital as opposed to just going to hospital. Any major childhood stuff, because that can again be relevant. And are you just ask, are your immunizations up to date? Again, always never be judgmental. And you can also ask if they've had something like this before, because a lot of time they have. And again, it just makes your life so much easier if you let the patient give you useful information. So you want to avoid jargonistic terms here as well. So like, don't ask if the patient has hypertension, ask them if they have high blood pressure. And that's another thing, sorry. So you can ask closed questions. So like, do you have any of these conditions? Something like that for like just really common stuff. So like asthma, diabetes, high blood pressure, that sort of thing. It can just be useful in case it's like they've missed something. Okie dokie, family history. So we usually ask like 3 generations. So we won't ask about children and grandparents for the same patient, just because it becomes less and less useful the further back we go. And make sure you actually check that they have siblings or children before you ask them how your siblings or children. And yeah, and if they're older, then like it can be good to ask if are your parents still with us? Are your grandparents still with us, before you ask about them? And it can also be useful just at the start to ask them like just a point blank question. Does anything like this run in your family, or do any medical problems run in your family? Because they probably will have a good idea of that. All right, we have a medications history. We use the mnemonic drugs. Do not tell the patient you're about to speak their drugs history, because although we of course understand drugs are like pharmaceuticals, it obviously has different connotations to the public. So one other thing that I do is that, this is me personally, you can do it as well. is I tend to do DUGS and then leave the recreational to the end, because people can be a bit more uncomfortable talking about like alcohol, tobacco, and especially illicit drugs. And so it makes it a bit more comfortable for them if they're like, oh yeah, well, they're just asking about all the medications. This makes sense. Like, I'm comfortable talking to this person about it. So what each of those means, so doctor prescribed, like, they've gone to a doctor, they've got a prescription, they're taking it. Recreational, alcohol, tobacco, or illicit drugs, and we do need to know about all of these, and we will be confidential, especially about illicit drugs. User drugs is like, you go to the pharmacy, you get Panadol, then you don't need a prescription, that sort of thing. Gynecological and hormonal, so like the pill, something like that. So the combined oral contraceptive pill, I mean there. Sensitivities and allergies, so anything that you know. And so make sure with sensitivities and allergies that you ask about side effects, because for one thing, like, patients can confuse side effects of like a actual drug with symptoms of an allergy. So they'll come and tell you I have a penicillin allergy, and then they'll describe the symptoms, and you'll be like, okay, well, that's just side effects of penicillin. Obviously that's still important, but you want to make sure, like, oh, do the symptoms actually match up with an allergy, or do they have some sort of sensitivity? You can mark it down, but like say, okay, the symptoms don't quite match up. And yeah, so if we identify any medications, they're like, oh yeah, I'm taking this drug and this drug and this drug. We want to know the name. We want to know how long they've been taking it for. We want to know the dosage, so how much are they taking? Side effects, that's a really big thing. Are you suffering any side effects from the drug? Because that might be linked to our presenting complaint. And compliance. So, people get prescribed drugs. People don't always take those drugs as they've been directed. So we want to know, excuse me, are you able to, so usually you use something like, do you manage to take those all the time? Remember, non-judgmental, because yeah, it's perfectly human to forget to take them sometimes, but it is important at the same time for us to know if they do. So alternative therapies, you'll probably hear them as like complementary and alternative therapies, so like naturopathy, acupuncture, something like that. Again, don't be judgmental about them. A lot of patients will use this, and it is important that we know them. In some cases, you might even have patients where one of these therapies is causing the symptoms. So you want to know about it. The patient is not going to tell you about it if you act judgmental about it. So just ask and present it the same as you would anything else. Okie dokie. So recreational drugs, this is again the big sort of sticking point. So you want to make sure you ask about it in a sensitive way, and also that you ask about it in detailed fashion. So alcohol, how often do you drink? What do you usually drink? Because, is it high alcohol content stuff? Is it low alcohol content stuff? And how much do you drink? Do you drink a large amount? Do you drink a small amount? So if they drink a large amount, maybe more than 2 to 3 standards a day, like consistently, then it might be pertinent to ask the CAGE questions. So CAGE questions are basically alcoholism screening. Like, is this person an alcoholic? And it stands for each of those words. So you ask them, ever felt the need to cut down, annoyed, blah, 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 blah. And if they answer yes to more than one of these questions, then it can be something to mark down and to report in your history. This is a potential risk factor. So tobacco, we ask, have you ever smoked? This is important to ask, like, have you ever smoked? Not do you smoke? Because, you'll ask the patient, do you smoke? No. Have you ever smoked? Yes. When did you stop? 2 days ago. Why did you stop? Because I knew I'd come in for an appointment. So we want to know, it doesn't really matter if they're currently smoking, if they have been smoking up until yesterday. So we want to know, have they ever smoked? And how long were you smoking, and how much a day? Because we can basically do something called a pack year history. So we say, like, how many cigarettes have you smoked? How many a day? And then we can convert that to a general, like a standardized measure of you have like a 10 pack year history or something like that. And that's useful for things like lung cancer calculations. And so illicit drugs, again, people are uncomfortable talking about this stuff, so you need to make them comfortable. So you'll say things like, this is a standard question we ask all our patients, because it is. You'll ask all your patients this. I don't care if they're a 4-year-old, well, not 4-year-old. You're not going to ask really young children about it. But you would ask, a grandma or something about it, because, hey, they might. Okie dokie. So social history, this is a big bank of questions. Like, you don't feel the need to like immediately memorize these. It'll become much more natural as you go. And there are a few mnemonics like essence that I don't have here, but which you'll learn as you go on, basically. So it's like a chat. Like, it's a lot of things, but it'll go quite quickly because you'll just, like, what are your current living conditions? Are you living in like a flat, an apartment, a detached house, something like that? Who are you living with? Are you living on your own, with a family, with a housemate, something like that? And then we want to know about their work, things like that. Basically, we want to know a lot of stresses because that can be quite relevant to disease. And then diet and exercise. Again, try to ask about these, like, do you have a good diet? Are you exercising well? Because those carry connotations. So you say, are you able to find time to exercise? Or what about your diet? How's that going? Because if you ask them that sort of questions, then you're more likely to get an accurate answer. And if you get an accurate answer, that's the best possible information for our history. Social support, that's sort of the other side of stress. Like, do they have people to help them through it? Belief system important to them. This is like obviously not super relevant to the main history, but it can be important to things like palliative care if we decide we need to do that sort of thing. And this one, especially recently, I'm sure you understand the implications. Have you traveled internationally recently or had contact with someone who's unwell? And this, even in non-COVID times, we'd ask about this for various other diseases. All right, any questions about the medical history, or the medical interview, I should say?
Did you just cover the whole semester?
Pretty much. Semester 1 clinical skill content is like quite sparse. Like, it's basically you do the history, and they teach you the intro 1 week, and then they teach you the more detailed stuff another time, and the next week, and then they'll spend the entire rest of the semester like doing the slight variations, like what if the patient's angry, what if the patient's sad, that sort of thing. So like what you need to know is basically this group of slides, like how to take this history, how to take it well, and then you'll get better and better as when you practice. So like I very much recommend, by the way, that you practice this because it'll become so much easier.
Yeah, sweet, thanks.
No worries. In an OSCE, so Will Browning asks, OSCE, should we take notes while the patient is speaking? Yeah, so in an OSCE especially, you want to make sure you take note. They're not going to be offended. Make sure don't be looking down the entire time. Like, try to make eye contact as much as you can. But like, you'll be asked to summarize back your findings, and like, if you're confident in your memory, and like especially if you take a history, you will usually remember most of the important stuff. But it can be useful at the same time to write stuff down. So you'll like usually develop your own strategies. So people usually have like a little section for medical history, for family history, for social, for medications, each of those. And then you could write down like WWQQAA, and there's all sorts of like shortenings and things and shorthands that you can use to make it easy for yourself. But yeah, like it'll come to you, you'll find a way that works for you. And as long as you remember to ask this stuff, you'll be absolutely Gucci. Any other questions? Okie dokie. In that case, I'll hand it over to Raymond to go over MedLaw.
Awesome. Let me pull up the screen. Just as a preface, this by no means like encompasses everything. There are baristas and lawyers that have like years and years of experience on med law. You're just getting a slight taste of it, and you'll probably meet lawyers and baristas who are doing med law in your tutes, and you can really ask them about the nitty-gritties of these laws and rules. To begin off, we have consent. So why is it relevant? Well, first of all, you need to get consent to perform any medical treatment on a patient, or you can be sued for negligence. And as a definition, informed consent refers to an individual's agreement to a procedure or treatment on the basis of having full disclosure of risks, benefits, alternatives, and consequences of the outcome. The gold standard acronym, I guess we always know, is VIC, which is voluntary informed consent. But and that involves being voluntary, so the patient can't be coerced into agreeing with it, informed in the fact that they know what the benefits are, the negatives, the risks, and the alternatives that are available to them before they say yes or no to the procedure, as well as capacity, which is the patient actually competent or able to make that decision? So they're not they're not psychotic, they have no mental illnesses, they're unconscious or under the influence of substances. And in terms of minors, some minors are deemed to be mature minors, which are teenagers who are under the age of 18, but they have the same ability to consent as if somebody who's above 18. This is usually assessed under common law, and there's a range of criteria that the person needs to meet, or it is deemed by the practitioner as this person is able to make is able to consent to the treatment. There are the three types of consent are implied, oral, and written. In terms of implied is basically, it is not explicitly given, but the patient's words or actions shows that he is consenting. For example, if you tell a patient you're going to do a blood pressure test and they hold their arm out for you to put on a blood pressure cuff, you can assume that they have given implied consent. Oral is basically clear agreement that is delivered verbally for a treatment. For example, if a doctor says to the patient, I'll be taking a chest x-ray, do you consent to it? Patient says yes, there we go. Written is basically for the more invasive procedures such as surgery, where there's a huge list of complications that can occur and you need the patient to consent that they are okay to proceed and they have weighed up between the risk and benefits of the surgery. And yeah, for example, anything that in neurosurgery, there's always a chance of nerve damage, and the patient must consent to that they may come out of the surgery with nerve damage. Basically, and then next is, when can consent be waived? And as most of us know, it would be in an emergency situation. For example, if a patient is unconscious, can I perform CPR on that patient? You can't really get the patient to wake up, tell you yes or no, and then perform it. So you just go with it. However, when the situation is not like critical or an emergency, and there's no life-threatening decisions that need to be made, the patient's consent must be asked for. Also to that is that if the patient is below the age of 18 and is not considered as a mature minor, usually you'd contact the parents of the child to get consent. Failure to obtain consent, this you'll explore a lot more during your actual classes, but in terms of, there's two forms of when consent is deemed invalid or not obtained, and that is battery and assault. With battery is basically the intention to contact with another person's body without consent. So basically, if you touch a patient without them, for example, a needle injection without asking them for consent to inject the needle, that's counted as battery, and it's can be counted as negligence, as well as there is assault, which is basically the less severe form of battery, as assault is basically the where you say something in terms that it creates a sense of that the patient will be harmed. For example, I will kill you, but although you're not actually doing the action, that fear that you can create with it is counted as assault. In this case, an example is when a doctor threatens a Jehovah's Witness to receive a blood transfusion. The patient now feels that the doctor will perform a blood transfusion without their consent, and they feel like they will be harmed, and that is counted as assault. When these two things happen, consent is not deemed adequate, and a doctor can be charged with negligence, which is when the doctor fails to meet expectations of duty of care. Issues of consent. Refusal of treatment. If the patient satisfies all the elements of consent and chooses to refuse treatment, then the right of refusal must be given. This sometimes comes into conflict with what a doctor wants, because sometimes a doctor might require a blood transfusion to save a patient. However, because they're Jehovah's Witnesses, they may choose not to, and you must respect that choice. Um, also, a patient may wish to not know the details regarding their treatment when they consent to it. And although a patient, if they say they do not want to be informed on it, that's all right, but you need to make sure, you need to insist that you will provide the information. But if they continually say that they do not want to know about the consent, excuse me, they continually say they do not want to be informed, the doctor should write this in the medical records. Disclosure, doctors are required to uphold doctor-patient confidentiality, and this is especially true in terms of disclosing any medical information about the patient. In cases where a patient's information can be disclosed are when the patient has consented to it, such as if the patient is like, I'm okay with you using this information to do X, Y, Z. If the patient's details are de-identified, so if you look up in a lot of journals, you'll see patient X who had Y something, something, so on and so forth, that patient has been de-identified, basically to the point that if some other person who may know the person who's in this journal, they won't be able to actually identify them. If it's in the interest of public health and safety, for example, if a patient has a communicable disease and they have a partner that it may result in harming them, you're allowed to disclose it, sharing it with the patient's healthcare team, as of course you can't help the patient if the other people in the treating team do not know what's happening with the patient, or if the patient poses dangers to themselves and or others. And then there are the legally obliged to disclose information such as a infectious disease. So for example, HIV or COVID or influenza, all of this stuff is reported to the infectious disease board. Medical malpractice, if it's required by the court or the police, reportable deaths, child abuse, and serious threats to public safety. Reportable deaths, some deaths are legally required to be for the doctor to report to the coroner, which is the person who will assess the body or the dead patient to see if there was any malpractice or fault or unconventional reasons for that person for death. So basically unexpected, unnatural, or violent or accidental deaths occurs during medical procedure as it may be a result of malpractice, or patients who are placed under the custody of Department of Justice immediately before their death, as malpractice or something may have occurred, an unknown identity, and any inpatient death at a designated mental health service. And then for the next part, we have sources of law. We have two sources of law. We have common law and statute law. Common law is basically rulings that are made based on cases involving similar legal principles. Basically, in Australia, the lower courts are bound by the decision made by the higher courts, and the highest court in the land is the High Court in Australia. And statute law is basically law that is created by the Parliament. Sweet. And that is med law in a nutshell.
Any questions for Raymond on med law? Okie dokie, I think we're golden. All right, thanks for sticking around. There's one more topic we have, which will go really quickly, and that's just the Australian health system. So bear with me one moment. All right, hopefully everyone can see that. All right, let me know if you can't see it at all, and I'm just presenting with a blank screen. Okay, so this is your HKS topic for week 2, the Australian health system. And this is basically just looking at how medicine in Australia works at a big picture scale. So this is really high yield. This is the 4 key components, and you'll come back to this in, for example, your HKS assignment. They'll want you to talk about these all 4 areas, and you want to make sure that you hit all of them. So we have stuff, technology, so our pharmaceutical drugs, our defibrillators, our MRI, CT scanners, X-ray machines, all of that. We need technology to treat patients, obviously. We also need people who are trained to treat patients. So our staff, doctors, nurses, paramedics, radiologists, all sorts of people are involved in treating a patient, and so all of them are important in some fashion. Spaces, so we need in GP clinics, we need clinic space there. In a hospital, we're going to need beds, ICUs. We need, if we have patients with critical illnesses, we need to make sure we have space for them, especially. And systems. So this is the last one. It's a bit more abstract, and it's talking about basically like how these organize. So we have 000. That's a service that will help you. They'll help connect patients to emergency services. We have our various like radiology, pathology units, that sort of thing, ambulance services, all of that sort of thing. This is also quite important, and we'll come back to this like just throughout the course, and also in potentially other disciplines as well. But the idea is we have 3 levels of healthcare, basically. So primary care is our GP stuff, basically. So like you go to the clinic, doctor, I've got a cough. Doctor diagnoses you with, a mild flu or just a common cold or something, and they give you something if needed, and they send you home, easy peasy. Now we can go up this pyramid by referral. So basically, if someone at the primary or secondary level thinks, okay, this is actually out of my hands, we need more specialist care, they'll bump you up a level. So doctor, I've got a cough. They see some things which look concerning, and they think the diagnosis indicates you need like specialist care, so you might go up to a respiratory physician. So secondary level care, so that's specialists. So like the 2 examples there. In our example with the cough, we've got a respiratory physician, so they're going to look at you. They're going to have a more detailed diagnosis, and they're going to have more treatment. So you go up there. It turns out it's COPD or something, which is a lung disease, by the way, and maybe that's the level. So like you have treatment for that. You set up a treatment plan, something like that. Or maybe it's something even more important. So let's say you have lung cancer. They need to operate on your lungs, and they need to exercise it before it spreads throughout your body. So they bump you up, they refer you to tertiary. So you'll have now a thoracic surgeon or something like that, and you'll be operating at a hospital and having an inpatient stay, something like that. So like as we go up the pyramid, we get more specialized and more rare. So you have a lot of primary care cases, some of those advanced to secondary care cases, some of those advanced to tertiary care. Okay, so it's important that we both manage the flow of these cases, and we also prioritize the important cases. So you don't need to know the minutiae of this, but like the basic idea is that like in Australia, we have a split responsibility between the federal government and the states in terms of who's actually responsible for doing healthcare. So the federal government is like Medicare, the Pharmaceutical Benefit Scheme, PBS, and funding of research. The state responsibilities is more like the nitty-gritty admin stuff, like funding public hospitals, licensing private ones, ambulances, other various little things. And then we have some shared responsibilities. So they might be both governments, or they might be non-governmental organizations. Those are up on the right. All right, and funding. So this is like I don't know how familiar you guys are with Medicare. I certainly wasn't familiar with how Medicare actually worked when I went in. It was just sort of, you go to the doctor, and sometimes you don't need to pay, and sometimes you do. So the idea is with Medicare levy, so that's 2% of your taxable income. And if you're a high income earner, they want to get you on private health insurance. So if you're not on private health insurance, they'll tack another 1.5% on top of that. And that's called the Medicare levy surcharge. But basically the point is that's where we get the funding from tax money. And then what actually happens is that money goes to fund free treatment for people at public hospitals. And from other providers like your local clinic, you'll get free or subsidized treatment. So you might have like, if you go to your local clinic, you'll see like a big sign at the front that says bulk billing on some of them. Basically what that means is the Medicare benefit schedule is a big document that says, here are all these things and like, procedures or drugs or whatever, and here is how much the government is willing to pay for them. And so your doctor can either choose to charge as much as the government will pay them. So they treat you and then Medicare pays them, and that's called bulk billing. So the patient pays nothing, but the doctor gets paid by Medicare. And you can also do beyond that. So if the doctor decides to charge more, then they'll get paid by Medicare, but there'll still be more money which needs to be given by the patient. That's called the gap payment. And that's very common in private practice, for example. The pharmaceutical benefit scheme is basically we want to have good access to drugs. We don't want poor people to be just locked out of medical care. And so on average for the drugs covered by the PBS, the government's going to pay about 87, almost 90% of our drug costs. And then the consumers are going to pay the rest. And we have various discounts for at-risk groups. Final slide. So systems thinking. So this is basically, you'll explore this a lot more in your tutorials, but basically we have lots and lots of moving parts in a health system. We have lots of different departments on any given patient, which are going to be relevant to which are going to be important to actually treating that patient. So when we think about systems thinking, we need to talk about all of the links between these systems and how to optimize them. So, we need to talk between these different groups. We need them to know what's important. We need to know which of these groups needs more resources. So the 4-hour rule is something that came from systems thinking. And it's basically, we have way too many people in hospital EDs, and we have heaps of backlog, things are not getting done, and high priority patients, like serious people, are not getting seen soon enough. So they decided in some hospitals to put in this rule and says, okay, within 4 hours, we're trying to get them out of ED. And so that's a good thing in most ways. So it helps us get more people out of EDs. We're not clogging up EDs. It helps us prioritize the important patients. So like, if there's someone who comes in and there's a serious condition which needs to be treated now, if we're in a hospital 4-hour rule, we'll have less people to look at and we can triage them and treat them more quickly. Obviously, that also has downsides. So if doctors feel like they're working to a schedule, then they might rush things. So there's always trade-offs for this sort of thing, but you'll explore that more on your tubes. And that pretty much finishes off. Any questions we have at all? Alrighty, in that case, I think we're more or less done. Thanks to all the presenters. Thanks to you guys so much for coming along and sticking around. And we'll see you next week at 10 a.m.